So good afternoon. In this video lecture, I will talk about the applications of PCR. This was the last part of my lecture series on PCR. I had made previous lectures about how PCR is done, how primer designing is done, what are the criteria for primer designing. We talked about RT-PCR, nested PCR, real-time PCR. And today we will talk about the applications of PCR. Why we do PCR. So let me write this. So we will talk about various versions of PCR here, how they are applied. The first application is we can say that the PCR is done simply for amplification purpose. When the amount of DNA that you have isolated from a sample is very, very low, say at the picogram level, then you may increase that amount, that DNA, to microgram level so that you can go for further analysis. Say you are studying, you are doing a fingerprinting of certain sample. And that sample is a blood spot or a semen sample. Very small amount of cells have been obtained from that place, and you have isolated DNA, and that DNA is so less that you cannot even do anything with that. So you need to amplify that. So for this purpose, number one purpose is amplification, simply. Amplification for DNA fingerprinting, or you may say RFLP analysis. Okay, so this is from the sample which has a very less amount of DNA. But when you have enough amount of DNA, you can use this PCR for gene cloning also. So the number two, you can apply PCR for gene cloning. So how this is done? You have to have a pair of primers. This is the criteria for cloning a particular gene using PCR. You require, suppose this is the long piece of DNA, and from here you want to clone this area. So you must know two primers of this region so that you can amplify that region and you can clone it into certain vector. So where from these primers can be designed? This depends on the information, the previous research that has been done. If you have a gene sequence of some related organism, you can use that sequence to design a heterologous primer. Or you can, if you have if you have protein, you are working with a protein and you have an N and C terminus, some amino acid of these two termini, N and C termini, known. So you can design degenerate primers from those amino acid sequences, and you can go for PCR amplification. It's true that the product of PCR that you will get and you will clone will not be a full-length gene in most cases. However, you can use that PCR product as a probe to screen full-length gene from genomic DNA library. So gene cloning is very important part of PCR. It is very much applied in gene cloning. Number three is you can apply this for disease detection that you are doing here in COVID-19, RT-PCR. So PCR can also be done. One thing, multiplex PCR is useful in detecting pathological diseases. Say in this case you can design 8 pairs of primers. And you can add these 8 pairs of primers in a single reaction. And these primers are of different organisms. They have been designed from the specific for different pathogens. So you know the history of pathogenesis or disease in a particular region, particularly the endemic diseases or sometimes some sporadic diseases. then you can design 8 sets of primer considering those diseases, those pathogens. If a particular person gets infected with certain pathogen within that group, then the DNA of the sample, the blood sample, the DNA that you isolate from that patient's blood will, or patient's sputum or whatever sample, will contain that pathogen, and if that pathogen is present, and if the pair of primers you have taken in this 8 pairs of primers, the set of primers, then that DNA will be amplified and you will directly determine, okay, this is the cause of the disease. So this is very useful and one-shot detection of the disease. This COVID-19, in case of COVID-19 also, you are detecting with the help of RT-PCR, because this virus is an RNA virus, so you have to start the process with RNA, reverse transcription, and then PCR. And this is also telling you about the load of the virus, the viral dose, the RT-PCR. This is yet another application, you can say separately, that RT-PCR is used to understand the RNA presence and absence of the RNA first of all, and the another RT-PCR, that is real-time PCR. See my previous lecture on real-time PCR. This gives you real-time information about the quantity of the RNA that is present in the sample. So how much RNA? You can study the expression profile of cells, of genes, of particular cell under particular condition. Okay. So number 5 is this PCR is applied for in taxonomic purposes in plants and in microorganisms, particularly the bacteria. Bacterial taxonomy is done. Suppose there is a technique called RDRA. This RDRA, amplified rDNA restriction analysis. This means you amplify the rDNA first, and then you go for restriction analysis, means RFLP. Then that particular, the binding pattern or profile is specific for a particular organism. This is what is RDRA is. Another is called RISA. This is a ribosomal interspacer restriction analysis. Suppose this is the rDNA, and here is the 16S rDNA sequence. And here is the 5S rDNA sequence. So if you take 2 primers from this region to this region, this interspacer region, the distance of the interspacer region is variable and it is specific for a particular organism. So if you analyze this, even if you don't digest it with restriction endonucleases, the size of this and the sequence of it particularly, it tells you about the organism. Okay, very specifically. In plant science also, agricultural science mostly, in case of rice, in fact, and there are others also. There are 2 techniques. One is called RAPD, rapid amplification of polymorphic DNA. In this case, you use several sets of primers and you amplify the DNA, and the amplification profile that you get is specific for a particular plant or variety of plant, in fact. You can screen this at the variety level. Another one is this AFLP, amplified fragment length polymorphism. In this case, you first restriction digest, and then you amplify certain fragments. And this also gives you partial information about the identity of that particular organism, that particular plant. We say that these are the principal applications of polymerase chain reaction PCR. These are also used in genomics research. Okay, I forgot to mention number 7, that using PCR you can create this STS, sequence tagged sites. These are the physical markers on the DNA. When you are doing, these are the sequence tagged sites are produced by PCR using random primers. Because when you are doing sequencing by Sanger's method, of course, not the new next generation sequencing, when you are doing sequencing with that method, what happens there are many gaps in the sequence, as well as you have to have smaller size DNA so that you cannot sequence more than 1,000 or hardly 1,200 base pairs. So to obtain smaller size fragments, you have to have a library, and that can be made by this, using random primers and doing a PCR and then cloning those fragments. Those are called sequence tag sites. And the other one that is called ESTs, expressed sequence tags. These are produced from the mRNA. These are one type of, you can say, cDNA, but they are also amplified by random primers. First, you definitely use the oligo-DT to do RT-PCR reverse transcription reaction, and then you use a random primer to amplify those. So these ESTs, expressed sequence tags, these sequences are specific for a particular ORF. So when you are reading ORF using some genomic tool, whether that ORF is the right ORF or not, it should be verified using these ESTs. If it matches with EST, it is an ORF. It is an active gene. It produces a particular RNA. Otherwise, sometimes some error occurs during ORF reading if you don't have these EST sequences. So these were the applications of PCR. Thank you very much.